Well, I'm just going to wait for a few more people to check in, and then we'll start on the meiosis lecture. All right. So, given that, hey, you can get to see my dog. I don't know if she's appeared yet in the. There she is. That's her favorite. I know, isn't she cute? Hang on, let's. It's going to be a little bit dim because the cats have run off with the uh, controller for the light and the fan. Little pests. I don't know what they've done with it. So I'm going to be like slightly in darkness. Let's see if I can move a little bit. Do, do, do. I'll lean over this way a little bit. Maybe I'll adjust. There you go. That's a bit better. Not that you really need to see me anyway. Um, all right. So first up, what I think would be worthwhile is opening the floor for questions about. Pardon me, cat. Quit it! All the cats are meowing at me. I don't know what they're doing. Anyway, where was I? Right, yes. So anybody got any questions about mitosis before we get on to meiosis? Good to get those out of the way first. Because today meiosis is the big one, so it'll be good to spend most of the time on meiosis if we can, but I think it's worth checking on mitosis first. So anyone have any questions on mitosis? Well, if you do, you can always just bring them up as we talk as well. Okay, so this is going to be a little clunky because thanks, Alyssa. I unfortunately forgot Angela, quick reply to your question. There's no lab recording or assignment for the Tuesday, what would have been a lab on Tuesday, because there was no lab on Tuesday. That lab was cancelled. I didn't update the schedule because I didn't forget, but I forgot to put it at the top of my list, and so I didn't do it, basically. But I hope that the announcements were sufficient. Yeah, so there is a lab this afternoon at the usual time. No, that's all right. And the materials for that are already uploaded. I think that's it. There's lab assignment was due Tuesday midnight. Yeah, that's right, from last Thursday. And the lab that's today will be due next Tuesday. Okay. Yeah, I'm not going to answer talk about lab questions now because we can do that in lab later. So, All right. So anyway, this is going to be a little bit clunky because, like an idiot, I forgot my laptop in Georgia. So that's currently on a USPS truck somewhere between there and here. It'll be here tomorrow. So I'm borrowing my wife's computer. So hopefully, fingers crossed, it all works. Okay. So let's get, if I can figure out. Ah, such a clunky piece of. Oh, no, I shouldn't say that. I'm very grateful to be using her computer. Okay, so, all right, so meiosis is a bit of a doozy, really. Hopefully you've already come across meiosis at least once in your Gem Bio 1 or Intro Bio 1 class. So it's not completely fresh, but there's a couple of big things going on. Oh, actually, give me just a second. Hey, Sov, you can have your phone back, love. For some reason, the internet works again. I don't need the hotspot. Here you go. Thank you, love. Oh, and our internet went out this morning as well to add to the fun. Okay, right. So Moses is, I don't think it's tricky to study, but there's a lot going on, and there's a lot of things that have to happen in a very particular way for meiosis to do what it needs to do. Okay, so there are two big things about meiosis. Yeah, two, generally speaking. The first one is the whole purpose. Well, one of the two main purposes of meiosis is to generate haploid gametes, right? So, sexual reproduction is essentially the fusion of one set of chromosomes from one parent with another set of chromosomes from another parent. I mean, fusion is not the right word. Fusion of two cells, each containing one set. So those come together, that creates a diploid zygote, right, which is like the very beginning of an organism. Single cell, two sets of chromosomes, one from one parent, one from the other parent, right? And so that in and of itself is going to create some genetic diversity because those two parents are presumably not identical. One hopes. Okay, so that's first big part. And so a lot of the various steps in meiosis and the two rounds of meiosis, right? Meiosis 1 and meiosis 2, their purpose is to go from one cell with two sets of chromosomes to, I don't have enough hands. I'd have to use my feet, right? Essentially, four cells, 1, 2, 3, 4, with one set of chromosomes, right? So there's two rounds of cell division in meiosis. One where the ploidy is reduced from diploid to haploid, the other one where sister chromatids are separated, right? And so in and of itself, that is fairly chunky in terms of material, right? There's a lot going on. Okay, so that's like the first big part of meiosis is to generate haploid gametes which can participate in sexual reproduction. The second part is kind of like to supercharge sexual reproduction, right? So the whole point of sexual reproduction is to generate genetic novelty. And the reason for that is that sexual reproduction is costly, right? If you think about it in terms of my genes are the best, right? Then in sexual reproduction, you're only going to give 50% of my genes to your offspring, right? So you're going to be diluting your genetic kind of heritage, essentially. If you were to produce asexually, like if I was just split myself down the middle and generate two of me, 100% of my genetic information would go down to each of my offspring. Well, kind of weird thing about offspring. I mean, each of me. Right? Anyway, so sex is costly, and also there are other costs, behavioral and stuff like that involved, like getting eaten by your partner, fighting over partners, things like that. So sex is costly, so there has to be some benefit to counteract that cost, right? Because otherwise you wouldn't do it, right? It'd be evolutionarily unfavorable. And that benefit is genetic novelty, right? And essentially, the purpose of genetic novelty is to cope with changing environments or new environments, potentially, right? If predation levels change or increase, or food availability decreases, or the makeup of the food changes, let's say, you don't have zebras no more, you got gazelles instead, then your offspring need to be able to cope and adapt to that change. And so if you're all identical, it's really hard to do that adaptation. Really hard, right? Because you've only got one set of instructions and information, and you can't change you, right? You can't change your own genetic makeup. So the purpose of sexual reproduction, because you want to separate those two. Purpose of sexual reproduction is to generate offspring which are varied and have different abilities to adapt to their environment, so that at least some of those offspring survive, right? Because obviously there'll be some that are more poorly adapted, but you hope there'll be some that are better adapted, right? And that genetic diversity or novelty is also part of the driver of evolution over time, because the ones that are best adapted are the ones that survive. So really, meiosis, in addition to generating haploid gametes, generates different gametes, lots of genetic novelty, right? And so there's a lot of randomness in meiosis, right? To be able to generate gametes that are as diverse as possible. Okay, let's see if I can figure out how to use this computer. Okay, so let's go over the kind of basis, and also if you have any questions at any time, let's see if I can bring the chat back up, make sure I don't miss anything there. Ask questions, right? Because essentially you're going to be tested on this, sometime today, right? And I want you to be able to do the best that you can. So there are two phases of meiosis. We're going to basically go over some of the kind of like the mechanics of it, right? So the 2 rounds of cell division, and also we're going to hit on some of that generating of novelty. Right, unfortunately, I didn't have time to fully go through this morning to refresh my memory. This is originally someone else's PowerPoint. Okay, so right, so first round of meiosis, called meiosis 1, is all about reducing the ploidy level, right? Going from diploid to haploid. And we do that by separating homologous chromosome pairs, right? So in meiosis 1, you're going to line up your homologous chromosomes. So this is like chromosome 1, this is sister chromatid 1, this is sister chromatid 2, right? They should be identical unless any errors were made in DNA replication, right? So this is one of a pair of homologous chromosomes, 2 sister chromatids. Here's another pair, one of the pair, another 2 sister chromatids, right? So this sister chromatid is not the same as this sister chromatid, right? These are non-sister chromatids because they're on different chromosomes. Well, different, yeah, it kind of get tricky. It's the same chromosome, but a different variation of it, right? Because it's a homologous chromosome. So in meiosis 1, those homologous chromosomes, that pair, are going to line up next to each other. If you remember from mitosis, all chromosomes pair up like so, right? In a line above each other, like people laying, feet to head, essentially. Meiosis 1 is different. Instead of being like so in mitosis, where you're going to be separating those sister chromatids, we're going to be lining up next to each other so we can separate the homologous chromosomes. That's how we get that reduction in ploidy. Then meiosis 2 is really just figuring out what to do with these sister chromatids. And so then the chromosomes lined up like so, and those sister chromatids are separated to either side of the cell. right? So meiosis one is called reduction or reductional division because you're reducing the ploidy level. Meiosis two is called equational division because essentially the ploidy level stays the same. Okay, come on, what are you doing? There we go. And so this is pretty much laid out here, right? So you go from 2n, right? N is the number of unique chromosomes, right? So we have 23 unique chromosomes, and 2 is the ploidy level. So this is a diploid cell. We separate, and these are the, I was trying to show with my fingers, kind of clunkily. These are the homologous chromosome pairs, right? So the N for this cell is 2 because you have two different chromosomes. You have this shorter one and you have this longer one, right? You have 2 of each, so it's 2n. So you got 4 total, right? It's kind of a good check. You can do the math. And so in the first round of meiosis, the first round of cell division, meiosis 1, and it's really important to remember meiosis 1, meiosis 2, right? Because you're going to have anaphase 1 and anaphase 2, and very different things happen in anaphase 1 versus anaphase 2, right? Same as metaphase 1, metaphase 2, so on and so forth, right? So you've got to be able to put that label, whether it's a 1 or 2, on whatever phase is occurring at that time. Oh, wow, my cat is yowling like crazy. I think she's just getting used to us being home again. So in that first round of meiosis 1, you go from 2n to n, or 1n. You go from diploid to haploid by basically separating those two homologous chromosomes into different cells, right? So now you only have one of each chromosome in each cell, and so you're haploid. And then meiosis 2 is splitting those sister chromatids apart, pulling those apart. And so really meiosis 2, other than the fact that these cells are haploid, or these cells, if you can see what I'm pointing at, supposedly there's, let's see, no, I've seen other people do fancy stuff with a pointer, but hopefully you can see what I'm pointing at. These two cells are essentially going to undergo something similar to mitosis. They're haploid, so it's not the same as mitosis, but the process is more or less the same. We're going to separate these sister chromatids so that each daughter gets one sister chromatid each. Does anybody have any questions about that? It's a good point to stop for a little bit. And shout them out, type them out onto the chat board, whatever works best for you. Okay. Oh, crab apples. I didn't get my coffee. That's a tragedy. No wonder I'm feeling so sleepy. Okay, so in oh, oh, thanks, Angela. That makes me feel a lot better. We got the same I got the same phases, right, as in mitosis. Got prophase. I mean, there's also prometaphase too, but it's simpler to think about in just these ones. PMAT, promase, metaphase, anaphase, telophase, right? And so you can see why there's one art and two after them, right? Because they're the same kind of processes, but what the exact details of what is happening is different. And some of those details are super, super important. Okay, let's move this over here a little bit. Oh, no, not that one. Trying to move this. Oh, well. Okay, so yeah, let's move these over this away a little bit. It's kind of hard for me to see them. Okay, so in prophase 1, right? So this is, so interphase of meiosis is, as far as I'm aware, exactly the same as mitosis, right? So the stuff that leads up to the division of DNA, the M phase part of meiosis 1, is exactly the same as mitosis. Now, some really different things happen in meiosis 1. Oh, I'm sorry. I mean, keep yawning. No coffee, you see? Tragedy. Anyway, so what we have here is we've got prophase split into 3 phases because there's a lot of stuff going on in prophase 1. And really, if there's anything you get out of this lecture on meiosis, is that meiosis 1 is where all the interesting stuff happens, right? Particularly prophase 1 and, oh, I got 2 thumbs, metaphase 1, right? Those 2 phases, I guess, are where almost all of the sexy stuff about meiosis happens, right? Or, particularly with regards to generating genetic novelty. Anyway, so prophase 1 is really involved, and the reason is that in prophase 1, those homologous pairs, right, they're lined up and they're actually stuck together, right? And they're lined up base to base, right? So if my, yeah, that isn't rude. If my fingernail is an A, right, on 1, it's going to be lined up exactly next to the one on the matching homologous chromosome, right? So base by base along, hundreds of thousands of nucleotides is exactly the same. And that's done by an enzyme, not an enzyme, a protein or complex of proteins called the synaptonemal complex. And I'm going to have to get a pen. Hopefully my laptop doesn't slide off. Oh, great. Cats threw all the pens on the floor. Yeah. Let's get these. It's all right, Doc. I'm not going to do anything to you. Okay. Let's stick this back. Okay, so yeah, let's get this out of here. Move that a little bit forward. So that's spelled like so. So synaptonemal, does that fit? There we go. Complex. They're talented and bloody noisy. Anyway, so this is really important because what this does is if you have your, ouch, that's kind of sharp. So that's one of your chromosome pairs, one of your pair of chromosomes rather. This will be your other one, right? Drawing them different colors just to illustrate that they're same but different essentially, right? It's kind of the best way of putting it. And so the synaptonemal complex, hopefully you can see, basically sticks those homologous chromosomes together, base by base, right? That only exists for a very short period of time, right? Only in prophase. That protein disappears after that because it has done its job, right? Does that make sense? It's purely there for the alignment of those 2 chromosomes, those homologous chromosomes. Okay, and so that happens really early on in prophase 1 to be particular, right? And so you can see that over here in early to middle prophase 1, those chromosomes are aligned and they're going to start condensing down, right? So they do the same condensation process or like the kind of scrunching up processes in mitosis, but they do it with the homologous chromosomes next to each other. Okay, so synaptonemal complex, remember that. Don't forget that the Shoot, I run out of colors. Should have brought more from work. The sister chromatids of each chromosome are stuck together with a different protein called cohesin, right? So let's just type that up if I can get to that. So a couple of definitions. Synaptonemo. If I can spell. Right? Whereas cohesin, oops, holds sister chromatids together in mitosis and meiosis. Right? So different things. It's important to get what they do because their jobs are different and they're both important. And as you know, if anything's important, I'm going to be asking you about it in an exam, right? Because I want you to know that stuff. Okay, so once those chromosomes are stuck to each other, right, they're going to do a second thing, which is also really, really important. It's important for a couple of different reasons. Let's see if I can draw this up here. So essentially, I'm going to draw them as if they're all condensed just because it makes drawing them easier. Oh, dog. Just got an ear infection again. All right, so that's kind of what they look like at the beginning, right? Once they've been lined up. Now what we're going to do is so this is these two, the green ones are sister chromatids. These two, the red ones, are sister chromatids, right? So the red and the green next to each other are non-sister chromatids. Oh, yeah, give me a sec. Is that better? Cool. I don't want to do that. Bugger off, thanks. Okay, so this green one and this red one are non-sister chromatids. These two are going to swap bits of themselves. So we're going to, the ones on the outside, however, do not, right? They stay the same, right? So this process is called synapsis or recombination or crossing over, right? All those three words apply. Right, so essentially we have the same, we're going to go from one to the other. Let's keep this lid off. God damn it, cat. All right. It's like a crazy house today in here. Now we're going to get those two kind of swapped over a little bit, right? So essentially what's going to happen is we're going to take that bit of the green non-sister chromatid and we're going to stick it on the end of this main part of the red non-sister chromatid. So after anaphase 1, you'll end up with something that looks, this is hard to draw and show at the same time. Put that like so. Something that looks like so. Right. And so you end up going from over here, where these sister chromatids are identical, through the process of synapsis, recombination, crossing over, whatever you want to call it. And at the end of, during anaphase 1, those homologous chromosomes get pulled apart. And now we have essentially two different sister chromatids, because this one is the original one from over here, right, or even over here, and the one that was next to the other one as they were lined up during prophase now has a different bit of chromosome from the other homologous chromosome attached to it, right? So this is one of the ways in which that genetic novelty is generated, right? Because again, remember, these homologous chromosomes are largely the same, but they're not identical, right? And because they're not identical, essentially these two homologous chromosomes, this pair, each one has a potentially different collection of alleles. So when you go through this process of recombination, let's say it's going to be hard to draw this on here, but let's say we have a whole range of genes on this chromosome, but we have different alleles on the different homologous chromosomes. So now we're going to get a different combination of alleles on these two recombinant chromosomes here than we did in the original parental chromosomes here, right? So actually we can at least do that. Kind of clunky and awkward, right? So these are what one would call parental chromosomes because they're the same as the ones over here, right? So these are essentially all parental, right? Because they're all the same, right? Same as each other. And these ones here are recombinant, right? So R equals recombinant because they're the result of mixing between those two non-sister chromatids. Does that make sense? Anybody got any questions? Hopefully this should largely be a recap from GenBio 1. Cool. Okay, so another important point to this is the following, right? So one purpose of this is to generate new combinations of alleles, right? We're not generating new alleles because that's mutation. We're generating new combinations of existing alleles. However, the second really important part of or outcome of synapsis is that, hello kitty cat, they hold together the homologous chromosomes until they're all aligned in metaphase 1. Okay, so remember we've got cohesin, right? Keeping these sister chromatids aligned, right? I'm kind of using different colors because I've got a fairly limited palette. Now, once you've crossed over, we have cohesin joining, yes, cat. These, she's very insistent, sorry. We've got cohesin joining together these sister chromatids, right? But now we have that join there, right? So essentially we have a bit of green, right, here, which is attached to this sister chromatid over here, is now attached to this red chromosome here, right? So if you can try and draw that up. Cat, get out of the... Oh, God damn it. Sorry, got to show you a picture of my cat. She's being very, very insistent that I pet her. Aren't you? Very fluffball. But she's kind of treading all over my markers and stuff. Yes. All right, so if we draw that in let's see where we got some space. Let's do it over here. Right, so essentially what you have is right, so that's the end, this bit up here, you can see it. This is the parental chromatid or parental chromosome to be. This is the recombinant one, and this one is stuck together with cohesin to its original sister chromatid, but also the new bit, this green bit, is stuck to its sister chromatid on the green chromosome, right? And vice versa for the other way around. Oops, wrong color. And I'm just going to draw them overlapped like that so that it's a little bit clear. Sorry, crappy picture, unfortunately, but hopefully it helps you understand. And so the point of that is in metaphase 1 of meiosis 1, there's going to be tension applied to those homologous chromosome pairs because we're going to have our spindle fibers, right, attached to either side, and they're going to be pulling them apart. So if recombination did not happen, let's say this stayed the way it does, as soon as tension is applied to those pairs, they will be pulled apart. So the fact that they are physically linked together by these chiasmata at the top and the bottom, there's usually one per arm, but there doesn't have to be. You could only just have one potentially. Because they're joined together, when that tension is applied to kind of pull them apart, the bonds of cohesin, particularly at the ends of the chromosomes up here and down here, hold those pairs together until the M phase checkpoint has been passed. Okay, so two big purposes from synapsis combination. One is to generate novelty, right? These new chromosomes over here, which did not exist in the parents. And the other one is to hold these homologous chromosome pairs together until the order is given for them to come apart during anaphase 1. Okay, so that's one big deal. Okay, does that make sense? Cool. Okay. And if it doesn't, ask questions because we don't get to recap this kind of next lecture before exam because the exam will start. Okay, so I want you to kind of get this. And so there's the really important point, another important point, they're all important, right? Otherwise I wouldn't talk about them, is that you can control what comes apart by degrading which cohesin in different parts. And so if you want to pull these, because essentially in anaphase 1, you want to pull these homologous chromosomes apart, or these ones, right? So that you get this, right? So they're separated. And so you do that by degrading the cohesin at the ends of the chromosomes, right? But you don't want these chromatids to come apart, right? Because that's what you're going to be doing in meiosis 2. And so you protect the cohesin around here, right, around the centromere from degradation. And so that means that the homologous chromosome pair comes apart, but the sister chromatids, or kind of sister chromatids, don't. Right? So that's how you get this at the end of meiosis 1, or end of anaphase 1. Yeah, I know, she's really noisy. Okay, you pet her and make her happy so she shuts up. Okay, anyway, kind of cornucopia of pets in this house. Okay, so you can control that, and the big deal is that because anaphase 1 is essentially promoted by the degradation of the cohesin on the arms, right? So that allows anaphase, which is when those homologous chromosomes get pulled apart. And so up here, and we're kind of bouncing around a little bit, so we're not really following the PowerPoint, but it's all in the PowerPoint. Around here, we have another protein, right? There's a lot of them, right? This one, I'm going to type it out for you, just in case you can't read my handwriting, is called Shugoshin. Don't ask me why. Actually, I've got to use both hands for this because my one finger typing is not very good. Right, so this protein, its job is to make sure that the cohesin that holds these chromatids together does not get degraded. Only but it does not protect the cohesin at the ends. So the cohesin at the ends of the chromosomes on the arms can be degraded by the anaphase promoting complex separase actually is the enzyme. The APC activates separase. So the enzyme that degrades cohesin on the arms, separase, let's write that down too, doesn't hurt. There you go. The separase that degrades cohesin here can't get at the cohesin at the centromeres because Shugoshin is protecting it. That's what keeps those sister chromatids together via the bonds that hold them, the cohesin that holds them together. But because the cohesin on the arms gets degraded, then you can pull those homologous chromosomes apart. Right, so as I said, there's an awful lot of stuff going on in meiosis. I mean, it's all like with anything, right? If you know all the stuff, it's all straightforward. If you don't know, it's really complicated. So hopefully this will help you understand it better so you can kind of put these things together when needed. Okay, so that all happens in prophase 1. And I'll stress that, prophase 1. Hello, KitKat. And the next really important part, so we've got one thing that produces genetic novelty. Right. One thing that produces genetic novelty is recombination. The other thing that produces genetic novelty is, and this is going to be an absolute bear to fit on here, even with my bad handwriting. Is random alignment of chromosomes or chromosome pairs in metaphase 1. You see all of that? Yeah. And so what that really means, and this is kind of cool because essentially, even though Mendel didn't know anything about this when he was doing his work on peas, this random alignment of chromosomes on the metaphase plate in metaphase 1 is essentially the law of independent assortment, right? So the law of independent assortment is what he came to by looking at the outcome of the crosses that he did, right? Of, 2 or more traits. But the cytological, the molecular basis of that is during meiosis 1. So essentially, and I'm not going to draw the recombination here because it would just take forever. I'm just going to essentially show you some different right. So these are really simple, right? We could do more, but it would just wouldn't illustrate the point any better, right? So this is our metaphase plate down the middle, right, where this kind of semi-imaginary line, right, the middle of the cell, right, equidistant from the poles. This is one potential way in which those chromosomes can align. However, the alignment of these chromosome pairs on the metaphase plate is completely independent from each other pair. So the orientation of this one has absolutely nothing to do with the orientation of this one, and it's random. Actually, almost everything in meiosis is random when we're talking about probability. Where those recombination events occur is random, right? And so you can see all this randomness. Fertilization is random too, mostly, right? All this randomness is what generates that genetic novelty, right? That's the point. So you could end up with, you don't need that one no more. That. And that, right? Equal probability. Or you could end up with that, also equal probability, right? So that means that every time you do a round of meiosis, right, from the same individual, you're going to get different gametes because it's totally random which way round these things align, right? And so in this case, we've got two different possibilities, right? And it's going to be, actually, there's another one I forgot, but I ran out of space. Right, so it's 2 to the 2, and so there's 4 different combinations. I've only drawn 3 up here, but there's another one as well. It's essentially this one, but the opposite, right? And so just from these, forget about recombination right now, right? Even though it is really important, and obviously we shouldn't forget about it, but in just simple terms, there are 4 different variations of this alignment that we can have, right? So just with that, we're going to have 4 different gametes for these two, for an organism with 2 chromosomes. Now, if we do us, right, we have 23 different chromosomes. I can't remember the exact number. That's how many different combinations we can get. So essentially, if you go through, you'd have to go through, just by probability, nearly 8.5 million rounds of meiosis, ignoring recombination, just thinking about alignment of chromosomes. You'd have to go through 8.5 million separate rounds of meiosis to get the possibility of getting one gamete the same. That's a really big number. Now, if you're, and we're talking about humans, if you're a male, maybe, right? We produce gametes all the time, right? Until we croak, basically. If you're a woman, you don't produce anywhere near this number of gametes. I think it's in the 500,000 range, give or take, right? While you're still an embryo. And so it's pretty much guaranteed that every single one of your oocytes, right, your eggs, if you're a woman, is unique just through this process of random alignment on the metaphase plate. Now, obviously, if you add to that the novelty generated by recombination, where these aren't actually the same every time, right, because of the swapping of bits of chromosomes, then this number just goes through the roof, right? And also another really fun thing, that's one side of the equation in terms of sexual reproduction. There's takes two to tango, right, when it comes to sexual reproduction. So if you times that by that, you end up with about 7 trillion something. So essentially, you, haha, as an individual, are 1 in 7 trillion. So your parents would have to have, you know, 7 trillion offspring to have the chance of getting another one just like you, which they're not going to do. You know, I like kids and everything, but I'm not going to have 7 trillion of them. Or at least my wife isn't. I think she has more of a say in that than I do. Anyway, any questions about that? Okay. So hopefully that helps kind of put into context why why meiosis is so important in sexual reproduction, and two, why meiosis one is really where all the cool stuff happens, right? Because there is obviously there's random alignment on the metaphase plane, metaphase two, but its consequences are much, much less. Okay, so let's get back to go away. This. Okay, so essentially at the end of so you're going to have these homologous chromosomes lined up. You can actually see in this image on anaphase 1, sorry, the different little bits, if you look real close, pardon me, of chromosomes that have been swapped over during recombination. And then you go through telophase 1, right? So you go through all the 4 phases, 5 if you want to be picky, that you do in mitosis, but you end up with a very, very different result at the end of meiosis 1. Now, some organisms just go straight into anaphase 2. Others go through a little thing called interkinesis. Let's see if I can. Right, we've already covered this, which is essentially kind of like rest and recovery between the 2 rounds of cell division, but no DNA synthesis occurs, right? All that needs to happen is the centromeres have to be, not centromeres, centrosomes have to be duplicated, right? So it depends on the organism. Sometimes some organisms during meiosis, there is a pause between meiosis 1 and 2 where, you know, chromosomes decondense and then recondense in prophase 2. In others, it does not. Okay, and so really, meiosis 2 is really simple. All it is separating those sister chromatids. They're not exactly sister chromatids anymore because they're not identical, but they're still attached together. And so they line up now end to end. So if you remember mitosis or, you know, feet to head, doesn't really matter which way you think about it. Those chromosomes are now going to line end to end because we want to separate the chromatids, sister chromatids, into different daughters. And so essentially that's what you get. So it's showing you this for essentially if we go back up here, right, this is the first round of cell division. It generates 2 daughter cells. The second round of cell division, meiosis 2, generates also 2 daughter cells for each of those 2. That's how we end up getting the 4. And so each of those daughter cells that was generated during meiosis 1 goes through the same process. So we have prophase 1, prophase 2, metaphase 2. Once they've all lined up on the metaphase plate, we have anaphase 2, telophase 2, and then we end up with 4 gametes. Now, this is a real simplified version of meiosis because the easiest way to think about this in terms of our context is this is what would happen if there was sperm. Right, this is how sperm are produced. Oocytes are they kind of go through the same thing, but the output is very different. In meiosis in human females, you end up with one cell, which is the oocyte that's going to be ready to be fertilized. And this whole process uh, occurs at different stages. So this is a really cool thing. It always fascinates me um, when I read about this. But essentially, if you're a lady, or even if you're a woman, probably a better term, right? All of your oocytes that are not yet activated, right, through your menstrual cycle, they are all maintained in metaphase one, right? So your oocytes have not actually completed meiosis, right? They're not yet ready. So when they get activated, and I can't quite remember the term, no, can't off the top of my head. A&P type stuff or animal phys is not really my strong suit. But anyway, once that particular cell is chosen for activation during the reproductive cycle, then it goes through meiosis one, and one of these cells survives. The other one becomes what's called a polar body, like the DNA stuffed into it, but nothing much else. And this is, again, don't stress too much about it. I'm just telling you this because it's cool, right? Not because it's like really important for you to know it. And then that one cell then divides again. I think this is, I'm just trying to think. I'll have to double check the details. Um, this is as it travels down the fallopian tube towards the uterus to be fertilized. I don't think it happens by fertilization. I think it happens just towards that, but also again generates what's called a polar body. So you end up, instead of 4 cells as you would in spermatogenesis, you end up with one cell in oogenesis, and the other two little bits are just kind of left floating around. Anyway, I just think that's cool. It's also just amazingly neat that in terms of human reproduction in females, all of your oocytes, all of your or oocytes to be, are kept in that state for your entire life. Obviously, once a month, one of them is activated. Um, but you start with, I don't know, 150,000, 200,000, something like that, give or take. Um, and so they're from before you were born, they've been paused in this state, right? And that goes all the way through until menopause, until you stop being sexually capable of sexual reproduction, which is kind of crazy. That can be like what, 50 years, 55 years, depending on the individual. And that's also why the risk of uh, aneuploidy, which is getting one too many or one too few chromosomes, such as in Down syndrome, which is trisomy of chromosome 21. The risk of that increases as you get older before, essentially before having kids, because those cells are kind of constantly in tension, right? And so there's a risk that they will, essentially that M phase checkpoint will fail. And one of those chromosome pairs gets pulled over to one side of the oocyte, and so you end up with an uneven division of chromosomes in anaphase one. So and the risk of that essentially goes up with time. So the older you are before having kids, the higher the risk. It's not a guarantee, but the higher the risk that the oocyte that did produce the egg to be fertilized didn't go through anaphase one properly. Okay, what else is there in here that needs covering? So again, 2 rounds of cell division. going from one diploid cell to four, potentially, haploid cells, right, called gametes, sex cells, germ cells, kind of whatever you want to call them. Gametes is the proper term, essentially, at the end of meiosis too. And there are two ways of increasing genetic novelty other than just sexual reproduction itself. One is recombination or synapsis or crossing over during prophase one. And the second is the random alignment of homologous chromosomes in metaphase one, right? And so those two things together, plus the random combination of gamete of egg and sperm, for example, creates that genetic novelty in the offspring. What else we got in here? Yeah. Oh, I was nearly right in the numbers. Okay, so this is, remember I was talking about earlier in the lecture, why those homologous chromosomes separate but not the sister chromatids, and the role of Shigoshin and cohesin in that. This is a good slide that recaps that. So if that kind of didn't make sense on the first pass, have a look at it here, and that should go through it in step by step, so you can kind of compare what I said with what's written here. And there's also a really nice diagram which basically shows that where the cohesin breaks down, right? So this cohesin along the arms, right, which is holding those two homologous chromosomes together via these swapped bits of DNA or bits of chromosome, that cohesin disappears, right? So that's what allows those homologous chromosomes to be pulled apart. But the cohesin at the center, because it's protected by Shigoshin, is not degraded by separase, and that gets you, but that only happens later. And I think that's pretty much it. I don't really have much more to say about this without just repeating myself. But I want to open up for any questions about anything. For the exam will be live in about 5 minutes, but again, you don't have to take it in 5 minutes. You got time to cover that. What I'll do is I'm gonna once we've finished talking, asking questions, I'm going to end this WebEx session so the recording is manageable. But I'll start another one, and I'll just keep that running while I'm drinking my coffee, in case anyone has any issues with the exam. Does anybody have any questions about anything? Exam, what we've covered today, what we missed on Tuesday, any of that stuff?
Are we meeting for a lab later today?
We are indeed, and that's at the normal time, which is 1:45. Yeah,
Okay, I just wanted to make sure.
yeah, 1:45. And all the material for that should be already in the folder for that, which is, oh shoot, was that lab 3? Corn and chi-squared, I think. Yeah. And also there were a few questions about the first, the first lab assignment. I don't think, I definitely haven't marked the second one yet because I was on the road yesterday. But we can go over any questions about the first lab assignment also at the beginning of lab 2. Yeah, good question, Sky. The first exam 1, which is today, will cover everything up to and including what we covered in this PowerPoint. So up to the end of PowerPoint 6. And Olivia, the exam is due today at midnight. So it will close at midnight, whether you've completed it or not. So essentially you have the whole day. You even have time to, if stuff occurs to you between now and when you start, you can ask questions and I can help clarify. If you have problems with the exam, send me emails. I'll be on my computer again most of the day. And yeah, you can start it whenever you want between now and midnight tonight. So that should give you a little bit of flexibility about, you know, going over anything if need be. Yeah. So I think that's pretty much it from the exam perspective. I think there's anything else that I wanted to say about that. Any questions about any of the material? No? All right. Well, I've got a coffee with my name on it, literally. So I'm going to go and grab that, and then I'll open up another. Um, well, actually, I'll probably just open this one again, uh, and I'll just be around. So if you want to chat or you have any problems with the exam or anything like that, I'll be here for the next, hour or so once I get my coffee. All right. Well, thanks very much, everybody. Glad you could all make it. And see you later for lab, if not before then. Okay, I'll do this. Dum dum dum. Okay.